All right, thanks, Leo, for the introduction. Yeah, really excited to talk to everybody today about what Anna and I have been working on the computational design of serine hydrolases. And yeah, let's get into it. Um, yeah, so the core sort of design principles or the core goal for enzyme design, I think, is somewhat straightforward, but of course over the decades, we've failed pretty constantly, and it's pretty easy to once you start thinking about it. But at its core, it seems pretty straightforward. How do we make an enzyme? How does an enzyme work? Let's just selectively stabilize the transition state and make the reaction go faster. So I sort of have this very simplified reaction coordinate shown here with energy on the y-axis and the coordinate on the x-axis. And for the uncatalyzed reaction, going from substrates to products, we have to overcome this transition state. And the goal or the role of the catalyst is to lower this energy and to make this reaction go faster. But of course, practically, there's a lot of other things going on to make this happen. The enzyme has to bind the substrate. Sometimes you have to proceed through an intermediate. Once you've generated the product, you need to release that product. So we have a lot of opportunities to fail, is how I'll put it. So you could fail to bind the substrate at all. Maybe there's no channel for the substrate to get into the active site, or you could bind the substrate too well and overstabilize this ground state and make it impossible to get over the transition state. The chemistry may just not happen. Your catalytic residues may not be positioned correctly, or you might just get trapped in an intermediate state. Some of these intermediates are pretty stable, and or there can be a conformational change that happens once you form an intermediate that prevents the chemistry from happening. And then sort of lastly in our list of failing possibilities is you could bind the product too tightly. So even if you are catalyzing the chemistry and stabilizing the transition state, maybe you bind the product with picomolar affinity and the reaction doesn't happen. So all this to say that enzyme design is hard, and trying to do it with a computer or rationally is definitely difficult because we have to account for all these different moving parts, in addition to, exquisitely stabilizing the transition state. And so we want to try to make the problem easier for us whenever we're doing computational enzyme design. So we use a model reaction. And the one that Anna and I picked, it's very similar to ones that people have been going for decades, and it's to go after ester hydrolysis. And so the substrate that we selected for this is an activated ester. So it's this 4-methyl umbelliferyl acetate, and we used a couple different acyl groups with this substrate. And the nice thing about this is that, one, it's an activated ester. So this 4-methyl umbelliferyl group is a good leaving group. And so it hydrolyzes a little bit more easily. And whenever it does hydrolyze, it forms acetate and then 4-methyl umbelliferone. And this is convenient for us because 4-methyl umbelliferone lights up when you're at the right pH. You get a fluorescent signal. And so we can easily track this, and we have a little bit easier time with the chemistry because it's more activated ester. Okay, so this is the reaction that we're going after. Sort of like I mentioned, there have been many people before us that have been making esterases for activated esters. And in fact, the first attempt on serine hydrolases was back in 1990. There was a paper about a sort of a simplified peptide. This ended up not being real. And people have tried with many other types of things that aren't serine. So cysteine hydrolases, histidine-based nucleophiles to break down esters, as well as metallohydrolases. And there have been successes with histidine, non-canonical versions of histidine that have a methyl group here. Probably a lot of the best successes have been with metallohydrolases. So Don Hilvert's lab, in collaboration with Brian Kuhlman, able to make some simplified metalloesterases combined with directed evolution to make some really efficient enzymes. But basically, we've been able to make cysteine and histidine and metallohydrolases, but for serine hydrolases, they've mostly just been failed attempts, which is sort of interesting. Why not serine, but all these other ones? And so here's just a list of reasons of why we think that this has been a difficult target for computational design over the years. One is that it's a pretty complex active site. So in terms of motif scaffolding, you pretty much at least have to scaffold 4 distinct residues. So we have the catalytic triad of serine, histidine, and glutamate or aspartate, as well as the oxyanion hole. So that's just a difficult motif scaffolding problem. The other thing is, serine isn't quite as activated as the other nucleophiles that was describing on that other page. So pKa of around 13.6, so it requires activation to get into its nucleophilic form. The other thing is it proceeds through multiple steps. So, metallohydrolases sort of have a benefit of going through a single step, whereas for serine hydrolases, you proceed through these intermediates, multiple tetrahedral intermediates and a fairly stable acyl enzyme intermediate. And that's sort of the last point here, is that many hydrolase efforts broadly that proceed through covalent intermediates, a lot of them have sort of gotten stuck in this acyl enzyme intermediate and failed to sort of do the second half of this reaction. So these might be the reasons that we've had a hard time making serine hydrolases over the years. And this will be helpful throughout the talk, so I'm going to step through the mechanism. The mechanism starts with histidine acting as a base to deprotonate serine, which acts as a nucleophile to attack the carbonyl carbon of this ester. This forms the first tetrahedral intermediate, and then histidine, once again, now acting as an acid, protonates the leaving group. This intermediate collapses to form the acyl enzyme intermediate. And so this first half of the reaction we call acylation. Now that we have this stable acyl enzyme intermediate, histidine shows up again, deprotonating water this time. Water attacks the acyl enzyme intermediate to form this second tetrahedral intermediate. Now, in the last step, histidine, now acting as an acid, protonates serine. This intermediate collapses, and we regenerate the enzyme. So quite a few intermediate steps to proceed through. Histidine seems to be a key player in this mechanism. So things to think about as we design these and characterize them. And so previously, and things that Anna and I did for quite a few years, was try to make these serine hydrolases with pre-existing backbones. And so what I mean by that is we would generate a theoretical enzyme active site, so sort of a model serine hydrolase active site. And then we would also generate a library of protein backbones or scaffolds. And then what we would try to do is match these active sites into these scaffolds, sort of find a perfect match so that we could scaffold our active site in a new protein. And we sort of like to think of this as, basically trying to buy a suit from a thrift store. If you just walk in and you hope to find a perfect fitting suit, it's unlikely that that's going to happen. And it turned out to be unlikely that we were able to find anything that perfectly scaffolded our active site either. So this approach, we were able to make activated serines, but we weren't able to generate serine hydrolases with this approach. And we think that this is because we couldn't, perfectly recapitulate all the features that are key to function. And so instead, can we make tailor-made serine hydrolases? So there's been this huge advances over the past 5 years or so in machine learning methods for protein structure prediction. And of course, now that's been used in a generative direction to make new proteins as well. So now with RF diffusion, we can start from an active site. This was sort of the dream before this existed. We give a serine hydrolase active site, and then RF diffusion can generate a protein backbone that scaffolds that active site. So this is sort of a tailor-made serine hydrolase, basically exactly the type of thing you'd want to be able to do. And so we used this approach, starting with simplified motifs of just serine histidine dyads. And the reason why we did this is because for initial motifs that we were using with RF diffusion, our in silico pass rates were exceedingly low. And basically what that means is that nothing was passing. And so what we decided to start off with is just a simplified version of the motif to increase our in silico pass rate. So we started with just the serine histidine dyad and then a single oxyanion hole contact that was coming from the serine nucleophile. We plugged this into RF diffusion, and so what you're seeing here is a trajectory of a backbone being generated around this active site motif. Then we use our sequence design tool, ligand MPNN, that assigns a sequence to a generated backbone. Now that we have a sequence, we plug that into AlphaFold to see if the designed structure aligns with the predicted structure. So does our sequence fold to our target structure? And these are our sort of filtering metrics that we landed on for AlphaFold filtering. And so in this first round of designs, here's an example design, REM 6507. You can see the serine histidine dyad sort of in a helical bundle fold. If you zoom in on the active site, this is what we're looking at. And we characterize these designs experimentally. So on the y-axis, we have fluorescence, and on the x-axis is time. And you can see that for the wild type or the designed enzyme, we're seeing an increase in fluorescence, but it stops around the enzyme concentration. And nicely, what we also saw is that if you knock out the serine or the histidine, this activity is ablated. But curiously, given that this sort of product formation is stoichiometric with the enzyme concentration, we suspected that rather than going through the full catalytic cycle, we were only going through that first half of acylation. So the substrate reacts with serine to form this acyl enzyme intermediate, but we're not able to hydrolyze this to regenerate the enzyme. But we were still pretty happy with this because we hadn't even seen this happen for about 3 years. So any activity was welcome, even single turnover. And it was nice to see that it's sort of over background, about 2,300 times faster than the background rate. So it seemed like, we were getting reasonable activity over background in this case. So that was exciting, but of course we want to be able to do full catalysis. So yeah, so basically we were wondering, how can we improve catalysis? There are many potential ways, and Anna's gonna start us on this first one.
Okay. Yeah, so like Sam said, we weren't happy with our single turnover enzymes. So the first thing we thought about in terms of improving them to make them full turnover enzymes was increasing reorganization. So reorganization is a concept rooted in the fact that proteins move. So proteins, the backbone's wiggling, the side chains are flapping around in the wind potentially. But something we know about enzymes is that enzyme active sites tend to move very little. In particular, the catalytic residues, which have to form these different interactions in order for the chemistry to happen, are always forming those interactions, even often when the substrate isn't present. And this is particularly true of serine hydrolases. And so we wanted to know if we could somehow evaluate whether or not our designs were pre-organized. And if you go to the next slide. So we had, luckily, a research scientist in our lab, Ivan Onishchenko, who was developing this deep learning model called Placer. And Placer took in a structural input. So for example, our design active site, and it would predict the conformation of the side chains and any small molecules in the active site. So it would sort of repeatedly corrupt or noise the coordinates of the side chain atoms, and then it would repredict those coordinates. And what was really cool about Placer was that you could run it over and over and it would be non-deterministic. So you could end up with many different conformations of the same active site. And we thought maybe this could be a good proxy for pre-organization. We could check if our enzymes are always occupying the same designed conformation or if they're also sampling sort of off-target, non-ideal states. And we had just screened all these designs for activity. So we decided we would just run Placer on the designs we had already screened and see what happened. And so we got different types of Placer outputs. So here are two examples on the bottom. We have some designs where every output from Placer looks basically exactly the same. So if we're looking at just our catalytic residues, serine and histidine are always forming the hydrogen bond that we want them to be. So that's on the left there. On the bottom right is an example of of a design where we do not always form this hydrogen bond that we want. Often the side chains occupy different conformations. And what was really interesting is that when we looked at which of these designs that we had screened did actually perform this single turnover catalysis, the two that we had found that were active, always in the ChemNet prediction, they were forming this hydrogen bond. And so we had this suspicion that maybe they were active and the others weren't because of this contribution of pre-organization to catalysis. And so if you keep going, yep. So we immediately added Placer as another machine learning technique in our pipeline at the end. So we would run through everything, AlphaFold our designs. Any designs that passed the AlphaFold filter would be subject to Placer filtering, and then we would only test designs that looked highly pre-organized and were forming all of these catalytic hydrogen bonds. And then another change we made was to the input motif. So Sam mentioned earlier, we had sort of simplified the motif so that it would be easier to generate designs that passed all of these difficult filters. But in nature, every single serine hydrolase has a catalytic triad and two oxygen and whole hydrogen bond donors. So we suspected that maybe this was something we would want to include as well if we wanted to make active serine hydrolases. So we spent a nice winter break generating many designs, running the computational tools for a long time, and came up with some designs to test. And very excitingly, when we tested this panel of designs, we did discover some actual serine esterases that were not single turnover. So this is one of these designs. It has a catalytic triad, a backbone serine amide oxyanion whole, and a glutamine sidechain oxyanion whole. And just like in the previous case, when we knock out any of the catalytic residues to alanine, we see complete loss, or any of the catalytic triad residues to alanine, we see complete loss of activity. However, when we see also a drop in activity when we knock out the sidechain oxyanion whole residue. So because these hydrolases continue to turn over, we're convinced that we had finally generated an active serine esterase. And you can see the catalytic efficiency and activities here are not great compared to natural enzymes, but we'll talk a lot about the efforts we made to improve these. So we were able to solve a crystal structure of crystal structures of some of these designs. This is just one of them. This design was like remarkably accurate. So our prediction was within 1 angstrom RMSD to the crystal structure. And when we looked at the active site, it just looked beautiful. We had this like atomic accuracy in all of the catalytic residues. all of the hydrogen bonds we had designed were formed in exactly the same geometry as we had hoped. So this was really exciting to see. And also interesting was that these designed serine hydrolases are really not similar to any known protein. We knew that they were really novel in terms of a serine hydrolase because they're all helical. Most serine hydrolases in nature are alpha beta or all beta proteins. But we kind of went overboard and we aligned these designs to every structure in the AlphaFold database. And our best alignment has a TM score of 0.46 for this particular design. This is like below the threshold for these to be considered the same fold. So these are very different. And also we know that another cool thing is that our active sites are really different from serine hydrolase active sites. So for example, in this particular design, it has a glutamine side chain oxyanion hole, which to our knowledge is not found in natural serine hydrolases. Okay, and as I said before, these designs are far less efficient than serine hydrolases in nature. So kcat over km of 22 per molar per second is pretty far off from, I mean, this is like the fastest serine esterase, acetylcholinesterase, which is a diffusion limited enzyme. And so, we couldn't point to any sort of small inaccuracies in our design model. This is basically as good as it can get in terms of design model matching the crystal structure. So the real question was, why aren't these good or even respectable enzymes despite being so accurately designed? And so we kept thinking about how to improve catalysis, and Sam's going to tell you about this next idea.
Yeah, so yeah, we were happy to make active enzymes. We were happy that they looked like the design model. But yeah, they weren't all that good. So, what can we do to try to improve catalytic efficiency? And so one thing we could do is try considering all these other states that are involved in the catalytic cycle. Seems like a good idea. And if you think back to our original design method, we sort of only consider a single conformation sort of throughout the design pipeline and at the filtering step. So we have this one input motif that in some ways represents the first tetrahedral intermediate or a transition state between the Michaelis complex and that first tetrahedral intermediate. And so we generate backbones for that, do sequence design for it, sort of AlphaFold the apo state. And then when we were doing these placer filtering things at the time, we were also doing that for the apo enzyme. And so could we incorporate some of these other states in that filtering step? And so one of the nice things about Placer, and now we have things like AlphaFold 3 and Rosetta Fold All Atom, that allows us to not only predict protein structures, but also predict proteins in complex with small molecules or even covalent intermediates or anything that's covalently modified. And that lends itself really well to sort of modeling the reaction coordinate for serine hydrolases. So we used Placer to predict not only the apo state, but also these intermediates along the path to get a sense of, is the design not only compatible with this one state, but is it compatible across the entire catalytic cycle? And so one of the cool things that we were looking at for some of these initial single turnover designs is just comparing them to some natural esterases. So we generated predictions for natural esterases as well as these designed ones. And so on the top, we're looking at the apo structure predictions. And so, the native looks great. It's got its catalytic triad fully intact. Our single turnover design looks not too bad in the apo state. But then if you look at the acyl enzyme intermediate, for the natural enzyme, again, it's perfectly locked in, and the acylated serines, sort of this carbonyl, Oxygen is perfectly positioned to interact with the oxyanion hole. But in our designed enzyme, you can see that there's actually a fairly significant conformational change. So there's both a serine rotameric shift, and then as a result of that, the methyl group from this acylated serine is sort of blocking any potential water attack of this acyl enzyme. So this sort of was giving us mechanistic, or at least a prediction that this might be the reason why this enzyme isn't multiple turnover, is that different intermediates are leading to conformational states that are unproductive, which is pretty valuable information that's very difficult to, or often very difficult to get experimentally. So it was very valuable for us to be able to predict this with Placer. And so what we set out to do is basically, can we take a design that already exists, but filter it over all the catalytic states, or even just part of them, and see how that affects activity or efficiency. And so in this case, we took a design called WIN. We did our sequence design protocol with like an MPNN, filtered with AlphaFold as we had been before. But this time filtered with Placer, either for both the apo state and the acyl enzyme intermediate, or over all states across the catalytic cycle. And we ordered and experimentally characterized two sets of designs for this sort of 2-state versus 4-state filtering. And we found that we were able to generate designs with improved catalytic efficiency. So WIN is shown here. This is the parent design. And then on the y-axis, we have catalytic efficiency. And yeah, about an order of magnitude, close improvement in efficiency. And then for sort of the best design in terms of efficiency was this one at 3,600 per molar per second, which in the world of designed enzymes that haven't been experimentally optimized is pretty good. One of the nice things about this too is that the sequence identity to the parent design is quite low, so we can do quite a bit of sequence redesign and still maintain activity, which is something that is often not the case for many enzymes. So we were happy to see that. We also determined the crystal structures of a number of these redesigns. And so again, they're pretty close to the design models. Some of them sub-angstrom overall C alpha RMSD. For each of them, the catalytic triad is still intact. And they're, in some of these, the oxyanion hole residue is also sort of fully intact, and some of the previous iterations of WIN, this would sort of move a lot or be in a different orientation. So we were happy to see that we were closely recapitulating a lot of the designed active site features. Yeah. And so, yeah, that's reaction coordinate. Anna will take optimizing catalytic sites. You might be muted, Anna. I think you're muted.
Sorry. Yeah, so this section sort of is based on the fact that there are so many natural enzymes out there, natural serine hydrolases that are really well studied. So people have done some really cool analyses about the different angles and geometries that we find between catalytic triads that are found in completely different folds. And so we were able to put our designs into that context and see if there were any sort of big differences between our designed enzymes and natural serine hydrolases that we could use to improve our own designs. So one of these differences that we noticed is in the angle of the second oxyanion hole residue. So it's been found that this residue is usually out of plane with the substrate carbonyl. And what we noticed is that our hydrogen bond from our second oxyanion hole residue is more in plane. And this may have implications in how well the enzyme can preferentially stabilize the transition states, these tetrahedral transition states that are forming throughout the catalytic cycle. And so what we set out to do was ask the question, if we move this second oxyanion hole residue angle to a more out of plane angle, could that be a source of potential improvement for our designs? So we had this particular design called Super, which had this in plane oxyanion hole angle. And what we did was we aligned it to a serine protease found in nature, cetylysin, which has an asparagine side chain oxyanion hole residue that's out of plane. And we just deleted the part of our design that held this old second oxyanion hole side chain and rebuilt the design so that it would scaffold cetylysin's asparagine residue in this new, hopefully better geometry. And so in the middle, you can see what that sort of looked like. We have in gray, everything that is still exactly the same as our original design. And then in green is the region that we deleted and rebuilt using RF diffusion to scaffold this teal side chain oxyanion hole residue. And so on the left, we can see comparison of the Michaelis-Menten plots between the original design and the optimized design. The optimized design has a much higher KCAT over KM. It's about a 20-fold improvement. That improvement mostly comes from KCAT, which is improved by about 10-fold, and KM is improved by about 2-fold. So, we can't say for certain that this comes from this very specific angle change because we've changed a lot about this design, but it is a cool kind of illustration of the way that we can use RF diffusion to really change different angles or geometries in our designs and kind of interrogate the effect of that. Okay, and then finally, the last idea we could come up with for improving catalysis was increasing the complexity. So our designed hydrolases, like a lot of design proteins, are these helical bundles. They have the full serine hydrolase active site, but hydrolases in nature are a lot bigger. They have much more interesting secondary structures like beta sheets and loops. And so we hope that we could tap into that space and hopefully improve our designs that way. And so why are all our designs helical? This comes mainly from this bias in AlphaFold single sequence prediction for helical proteins. So AlphaFold is just better at predicting helical proteins without MSA information. So we would make these thiazymes that had, small helix chunks and beta strand chunks on the catalytic residues. So we had about 60% all alpha thiazymes, or these input motifs to diffusion, 40% alpha beta. We would run RF diffusion and we could look at our diffusion backbones and what kind of secondary structures we were seeing. Similar, basically exactly the same proportion of alpha and alpha beta diffusion outputs. But then when we would run these through AlphaFold, we would lose basically everything that had beta character. And we would just end up with a lot of all alpha designs. And then when we would run ChemNet, we would lose also a lot of designs that were deemed not pre-organized by ChemNet. So in the end, we would test very few beta-containing proteins, and this was a little bit aggravating because all serine hydrolases in nature have at least that I know of, have at least some beta strands. And so we asked this question, like, how can we bootstrap AlphaFold to predict these more complex topologies from single sequence by sort of giving it a little bit of information about the design? So what I mean by that is we can give sparse information in the template to AlphaFold, in the form of these contacts. So we can tell it the distance between pairs of residues that are far apart in sequence. And maybe that small amount of information, if we just give a few pairs, will allow it to then figure out what the structure is. And so what we're seeing in the middle, this green line here, is basically if we take a single design and give it more and more of these pairs, these contacts, we can see how AlphaFold's prediction compares to our design in the RMSD, and we can see that RMSD at around 20 contacts just suddenly drops to the exact match to our design. So AlphaFold is like, even without any information, is not that far from predicting our structure. It just needs a little bit of help. And so we thought we could maybe use this to get a little bit better of a pass rate for these more difficult topologies. We didn't want to give the entire structure to AlphaFold because that's kind of cheating, but we still wanted to preserve some of the filtering power of AlphaFold by giving it just a few contacts so that we would get a passing rate that we could work with. And so Kira Sumida in our lab, she's a grad student in our lab, did a lot of this work designing these proteins with these serine hydrolases with these like more complex topologies using this bootstrapped version of AlphaFold. And she was able to generate these designs that are our best designs that we've been able to make so far. KCATs on the order of 10 to the minus 1, and KCAT or KMs in the thousands per molar per second. So we were really excited about this, and yeah, we were happy to see some sort of more respectable enzymes being generated. And there's still a lot of room for improvement. So again, here's acetylcholinesterase, which we are battling against KCAT greater than 4,000 per second. And we were still around, I don't know, 10,000 times slower, 20,000 times slower than this. So there's still this question of what is missing. And we have one more idea that we haven't looked into too closely, but it could be the source of another source of this discrepancy. And this is sort of in the way that we're designing these proteins. So we're using ligand MPNN or protein MPNN to design a structure or a sequence given a structure. And so protein MPNN is really giving like the most probable sequence, the best amino acid at every position for that structure. But we kind of know that in natural enzymes, this most probable residue is usually not the most functional residue. So even at a very basic level, we know that enzymes have these pockets that are largely hydrophobic, but then they have to contain these polar or charged residues, which would be sort of unfavorable sitting in that hydrophobic area, but they need to be there in order for catalysis to occur. So we kind of know that there is this discrepancy between probably what protein MPNN or ligand MPNN wants to give us as our design sequence and this like functional aspect that we obviously want to achieve in our designs. And so this is maybe something that we will keep thinking about, how to optimize our deep learning sequence tools to give us more functional enzymes. But overall, we think this approach is kind of a powerful new way to do enzyme design. So like Sam said, this is kind of how we always wanted to do enzyme design, but we just didn't have the tools. So we can start with an active site, build a scaffold that is tailor-made for that active site using RF diffusion. And then we think this tool placer is going to be really important for filtering designs and deciding which designs are the most pre-organized, which designs are adopting the right geometries for catalysis. And so here we presented this design of serine hydrolases, which already exist. They're one of the most common enzymes in nature, but hopefully we can use this approach for designing enzymes that do novel chemistry or use mechanisms that we don't have a handbook for already in nature. And so, yeah, we want to thank a lot of folks who helped us out with this project. In particular, Kira designed many of the enzymes and the fastest enzymes that are in this work. Yvonne developed Placer. David Jurgens developed the flavor of RF diffusion we used heavily in this paper, and many other people in the lab. We also want to thank the staff at the Institute for Protein Design who are really instrumental for keeping everything running, and David Baker for guidance. And yeah, thanks to Leo for the invitation to talk at this seminar, and thanks everyone for your attention. We're happy to take questions.